Please post that down in the chat. Thank you so much, Soundarya, for sharing, and also everyone else. Okay, great. That is great to meet everyone. So moving on, my name is Deepsha Biswas, and I am the Omixlogic community manager responsible for daily interaction with thousands of users who are part of our growing bioinformatics community. So as you see here, we have a lot of participants who join us from everywhere in the world. And even in today's session, as you see that all of you are coming from different places. So this is a great opportunity to not only learn from a session, but also interact with each other and learn from each other. And now, moving on, I wanted to introduce you to the OmicsLogic program, the OmicsLogic Transcriptomics program. We had organized this twice before this year. So this is going to be the third time that we will be doing this program. And we had many participants who were able to successfully complete the program. And they were undergraduate students, graduate students, faculty, and PhDs. And the feedback which was shared by our previous participants and their progress is very encouraging to our team, as well as to the participants who will continue to take these courses. And the main thing is the experience in designing bioinformatics projects with the help of mentor support, which is an important aspect of our OmicsLogic programs, which has helped many of our previous participants. As you see here, Urja was a participant in our research fellowship program, and she completed a project on differential identification of biomarkers between neural stem cells and glioblastoma stem cells. And then she went on to win an award for doing this project, which was an award in a student research congress. And like Urja, there are other participants who had participated in different team challenges, won different awards, and continue to do this. So these are all these photos or screenshots that you can see that you will be able to see even in our LinkedIn channel from different participants who share this. And now I would want to invite Ilya Brodsky to speak more about today's session and introduce you to NGS for gene expression studies. So over to you, Ilya.
Thank you, Deepsa. Okay, so welcome everyone to this webinar. For those of you that don't know me, my name is Ilya Brodsky. I'm the co-founder and CEO of Pine Biotech. What our company does is that we really have a vision to enhance human health and well-being by enabling biological research and discovery, providing relevant data, solutions, and support. Our mission here at Pine Biotech is to simplify bioinformatics, make it more accessible, helping different types of users participate in advanced research. One way that we are doing this is through our modular and intuitive multi-omics analysis platform that we will talk about today. But importantly, we appreciate the role that training, research support, and big data analytics tools that are designed for different types of researchers and different areas in biomedical discovery. So our mission is really to support cutting edge research and to solve the big data challenges. We want to make sure that our solutions are easy to use, and we engage in these training programs so that those that are interested to use big data analysis tools can use them effectively and arrive at sound and reproducible results. So while we focus on research support and develop tools for research, we also do a lot of training. And this program is an important program because we see many biologists struggling with the types of data and the types of insights that these data can provide that are driving research ideas and helping them develop more competitive and more compelling research projects. So what this program in transcriptomics will do is introduce RNA-seq processing, so you really understand what the data represents and where it comes from. It will also allow you to try different tools that prepare the data for analysis, explain some of the statistical principles in analysis of transcriptomic data, whether it is a simple experiment with just a couple of conditions that could be compared, or there's a more complex experiment design, but also importantly introduce you to machine learning tools that are commonly used for analysis of big data sets. As you know, these big data sets have been increasing in their complexity. And so to illustrate how some of these methods are used, we're going to take a project-driven approach that explains exactly how these different aspects fit together into the analysis process. So as you know, next generation sequencing is now a technology that is well accepted and has become very affordable and accessible, providing huge consortia that give us access to thousands of samples on any given type of a phenotype. And so we'll talk about how phenotypic data that could be illustrated by cell morphology or tissue type could be further explored at the genetic level, where we see genetic variation at the DNA level, different aspects of epigenomic regulation. And then transcriptomics, what this data provides us with is an ability to infer the relationship between what is encoded in the DNA and what we see produced at the protein level. So at that level, it is very interesting to observe kind of a snapshot, a molecular detailed snapshot of what's going on inside tissues, cells, and individual organisms. So what we'll do in this program is we'll talk about how, as a method of measuring RNA translation, transcriptomic studies can help detect mRNA expression inside the cell as an accurate alternative to membrane protein expression detection, which allows for molecular precision in diagnostics and new insights for the development of treatments based on molecular pathways. So RNA-seq as a methodology has revolutionized the way that biologists examine transcriptomes and has been successfully applied in many areas of basic biological research, drug discovery, and also clinical diagnostics and treatment selection. Compared with other technologies like microarray-based transcriptome profiling, PCR quantification, RNA-seq has a much wider dynamic range, which means that it can detect many variations of the same transcript alternative splicing, avoids a lot of the technical limitations such as varying probe performance and cross-hybridization. And RNA-seq can measure the expression levels of thousands of genes simultaneously and provide insights into functional pathways and the regulatory networks in biological systems. Importantly, RNA-seq can provide novel insights into alternative splicing, detect unannotated exons, and novel transcripts. Most importantly, such detail has already proven to yield significant discoveries in many research areas. So what this program will focus on is to explain how bioinformatics is an important component of such studies and is designed around integration of multiple types of data, transcriptomics being the primary focus, but nevertheless looking at the wider picture of how we can understand complex conditions such as diseases at the subcellular level and identify important players in this network of associated features. How analysis of these interactions can help us identify novel patterns, insights, biomarkers, and targets for drug design and repurposing. So biomedical projects that we will use will show how such aspects of diagnostics could be applied to cancer biology, how we can classify different diseases and stratify patients, understanding different groups and associations between biomarker features and patient outcomes. And we'll also look at single cell and tissue composition. So the program is really designed to give you a high-level overview of bioinformatics, specifically focus on transcriptomic data analysis, and then introduce you to some resources like projects, training materials, and tools that will help you make the most of RNA-seq data. So let me briefly explain how these different topics are going to be covered in the upcoming program. We will start with an overview of tools and resources. We will then go to speak about how to process next generation sequencing data that comes out of the sequencer. And we'll talk about what are bioinformatics pipelines and what are some of the design strategies for these pipelines to make them produce insights that we can later on analyze using downstream analysis tools. Then we'll talk about preparing the data for in-depth analysis. It's important to understand normalization, how the data is distributed, what are some of the technical variations that we should eliminate before we try to compare different conditions and try to infer biological meaning. We'll then talk also about a new, I think, paradigm in such data when we're going from just a couple of samples per condition to really tens or even hundreds of samples that allow us to train machine learning models and design predictive analysis to really understand association between clinical and phenotypic classes and specific molecular features that we have in our data. We'll then talk about another emerging technology, which is single-cell RNA-seq data. And there we'll look at tissue-specific expression based on analysis of thousands and even millions of cells at a time, where we can analyze specific variation and compare that variation by tissue, cell type, and association with different phenotypes. The ultimate goal of this program is to help you design a project of your own. And so we will provide you with several examples, but we will also allow you to try to find and design a project of your own. And to support an in-depth research project, we will also introduce you to a variety of tools. But importantly, we will guide how that process happens from the start of selecting your data to then designing your analysis, and then finally working through all of the technical issues that are associated with that. And to help you make the most of that, we have a number of mentors that will be helping to go through that process with you. And some of those will be speaking today as well. Now, the process of this whole program is designed around a schedule that you can see here on the screen. Starting from December 8th, we will meet 2 or 3 times a week for 3 weeks, but the whole program is designed around a month. And during this month, what we've seen in the past is you would be surprised how much progress you can make as somebody who is completely new to bioinformatics, or as somebody that knows about some of the technical aspects of bioinformatics, but has never really applied them to your own research project. So that's really the objective of this program. Now, this program consists of several elements that are built to work together to help you make the most out of the program. And that includes interactive sessions where we meet in a group setting, go over the material, answer questions, and try some practical assignments together. But in between sessions, you will have access to self-guided study materials. And those materials are designed to break down the complex topics that require some in-depth review additional research materials that will help you understand the problems that we are solving, and also some practical assignments that are going to be done both using an intuitive platform that I'll show in just a moment, as well as some practical assignments in coding that are optional, but will help you understand what goes into producing different types of data structures, visualization of those data structures, different types of analysis techniques, and biological interpretation. So the online coursework is designed to help you study independently and will help you prepare for the different interactive sessions to make the most of them. During those asynchronous materials, you will be able to watch videos, review the text, and really understand the details behind some of the things that we are speaking. Now, in these courses, you will find important information that will help you keep track of important words, understanding some of the research reasoning behind what we are discussing, and really understand how all of this fits together. So it's a very methodological process of going through the material that if you already know, you will be able to kind of skip over. Now, you will be able to download all of these materials as PDFs and make sure that you understand all of these and can have them for later as well. So as I mentioned, there are also practical assignments, and this is an important part of bioinformatics, that we don't only speak about how bioinformatics is applied, but we actually try to do it ourselves. And so here you will find specific types of reviewed assignments that you submit and get feedback on, and also get to try some of the tools, both as an interactive platform that I will show in just a second, but also you will see how the platform helps introduce you to the details of the methods that we will discuss. So let me show you briefly what that really will look like. For those of you that want to follow along, you can go to server.tbio.info. So I'll just put the link here in the chat. Thank you, Bipsa. So let's go over to this platform, just so that you have a really clear sense of what we are going to be doing. So this is the tBioInfo platform. You can see server.tbio.info. The platform allows processing, analysis, and visualization of multiple types of omics data. So that's why we are talking about a multi-omics integration logic in a lot of our programs. This logic is circled around processing tools and then downstream analysis tools that I'll talk about in a second. So for transcriptomics, we will explore how you can analyze specific types of output data from a sequencer, which come in a format that contains short sequences of reads. Then those reads are processed to eliminate technical variation. And after those are trimmed out and pre-processed, you can proceed to go on to mapping to the reference genome. Now, that could be done using several different strategies. It could be a detailed view of alternative splicing events. It could be a mapping on the transcriptome, or it could be what's called a joint strategy. So a strategy that combines both mapping onto the whole genome, preparing a novel transcriptome, and then mapping onto that transcriptome. Now, these are different strategies that are important to understand because they play a role in our ability to ask specific questions of the data and have accurate information for us to be able to validate our assumptions based on what we find in the original data that came out of the sequencer. So we'll talk about some of these pipelines. We'll also talk about some of the variation of the pipelines and some of the downstream analysis, where you will find such popular packages as DSeq and others. So importantly, in this process, you will be able to understand the logic behind what we did, understand how each one of the methods is addressing a practical challenge, and then see the results for yourself in the context of a project, right? So really understanding the biological, clinical, and research significance of a project data set. So as you go through this process, you will find that some of the methods provide very standard outputs, and those outputs have to be expanded on. So you might want to modify the way that you visualize, maybe change some of the settings. So you really need more flexibility. And to provide you with an insight on how to address some of those questions, we have designed some practical assignments that you will see as we go through the program using coding exercises. In these coding exercises, you will get an introduction to methods of using Python and R, BioPython, and different packages in R, to really go through some of the downstream analysis, be able to load your data and visualize your results. So just to, again, illustrate what those will look like, I'm just going to show you a quick example in R. So in this program, we will, for example, focus on differential gene expression. And so after you've learned about differential gene expression, You will get a breakdown of how the data is loaded in and prepared for analysis. After you've gone through that, you will be able to run your analysis right here in the browser. And so what's important here is that we will go through some examples that have variations at the level of the input data that you see right here. But running this in the browser provides you with a problem-free and bug-free environment where you can get your results, and we can all do this in the context of a single session. So you don't only get to learn about the code, you also get to run it, and we get to compare results at the end. So as we talk about this program, as I mentioned, the tools are a critical, important part of this program. And they are not designed for just people that know how to code or for bioinformaticians. They're designed for biologists and clinicians, so anybody can learn from this experience. After you've learned how to prepare the pipelines and you understand each individual method, you'll be able to produce outputs that explain what the data shows from a biological perspective. So we'll go through stages of statistical analysis, but also downstream interpretation in terms of pathway annotation and in terms of our ability to think of the biological implications of the variation that we found. But importantly, we'll go through an in-depth view because many of the biologists don't really understand what goes into preparing the data and how variation in the processing pipelines plays an important role in your ability to make certain assumptions and interpretations of the data. So we'll talk about many of the important steps of pre-processing, understanding the data, being able to do exploratory analysis as well. To help you go through that, it will not be enough to just be in the sessions. You really do have to go through the synchronous materials to get all of that completed on your own. And here I want to say that also for those that cannot attend the interactive sessions, we are providing these materials as a separate resource that you can get access and do them on your own time. And I think that going through the explanations and also doing the practical assignments will help you understand what goes on to understanding your data and then trying to work with that data so that it makes a lot more sense once we get to data mining and classification. I think that's where a lot of programs skip over the basics to go to the more interesting things. And I think that, big data has been playing a major role in how research has been changing over the past decade. And here, the introduction of machine learning tools have transformed our ability to deal with the emergence of these comprehensive data sets that allow us to really ask complex questions and get predictive answers about some of the conditions that we have. So in this program, we'll talk about a lot of the typical approaches for supervised and unsupervised data analysis using data mining and classification, really understanding how the different methods allow us to ask different types of questions and provide us with different types of outcomes. And as I mentioned, these outcomes are going to be based on biological examples, and we'll go from a high-level introduction of the logic all the way into being able to code this kind of analysis on your own, starting with the code playground that I just briefly showed, and eventually going on to try to apply those on your own system if you wish. So really, it's a combination of a variety of tools and resources. And hopefully, many of you that join will find that an intuitive experience because it connects all of these resources together under your own profile. And at the end of the session, Vipsa will go through some of the stages of registration and preparing your profile so that you can make the most out of this experience. Now, one of the people on our team that will be mentoring you throughout the session is an experienced bioinformatician. She has a PhD in bioinformatics, and she will be guiding you through some of the materials that are a part of this program. So I want to introduce you to Dr. Harpreet Kaur. She is an OmicsLogic trainer today, but she recently did her PhD with several publications focusing on liver cancer. In her current research, she continues to apply machine learning tools and multi-omics integration tools to really understand the biological drivers of this disease. Harpreet, let me pass it on to you.
Thank you, Ilya. Let me share my screen. Hello everyone, my name is Harpreet Kaur and I have a PhD in bioinformatics. My research is on biomarker discovery for diagnosis and prognosis of liver cancer. So what inspired me to study liver cancer for my doctoral research is the high mortality rate of this malignancy and lack of precise biomarker that can help diagnose liver cancer at an early stage. Worldwide, liver cancer is considered as sixth major cancer in terms of incidence and fourth in terms of mortality. It is often at times misdiagnosed or detected too late for an effective treatment. Another factor that contributes to poor outcome of the liver cancer patient is the frequent reoccurrence of the tumor. So with these concerns in mind, I have framed number of research objectives with my collaborators to address these issues using various bioinformatics techniques. For the first part of this research project, we used large-scale transcriptomics data to identify a diagnostic biomarker panel for hepatocellular carcinoma, which is primary liver malignancy. So in this study, we employed 30 data sets containing nearly 4,000 samples to elucidate transcriptomics biomarker panel for diagnosis and prognosis of liver cancer, implementing various statistical and bioinformatics techniques. For the next part of this research project, we used transcriptomics and methylation data to identify a biomarker panel for stage classification of hepatocellular carcinoma. Here, we employed TCGA data of 350 samples and elucidate 51 features that include 30 RNA transcripts and 21 methylation CpG sites using a number of data mining techniques. The naive-Bayes model classified early and the late stage sample of HCC with nearly 79% accuracy. For the third part of this research project, we used transcriptomics data to scrutinize diagnostic biomarker panel for cholangiocarcinoma, which is another major liver malignancy. So in this study, we employed 9 data sets containing 350 cholangiocarcinoma, 133 adjacent non-tumorous, and 90 HCC samples to scrutinize cholangiocarcinoma specific transcriptomic signatures, implementing a number of data mining techniques like extra tree, SVM, random forest, logistic regression, LDA, etc. So in conclusion, during this whole research project, we identified a universally platform independent 3-gene based biomarker that can predict HCC samples with high precision and also possess prognostic potential. Then we identified a 3-gene based signature that is specific to cholangiocarcinoma and they are capable to discriminate CCA from HCC in the normal samples. At last, we identify 51 features that include 30 RNA transcript and 21 methylation CpG sites that can predict HCC at an early stage. So in my current role, I'm OmicsLogic bioinformatics trainer with Biotech. I'm assisting participants to learn about data analysis, machine learning, and data visualization in application to large data sets used in biomedical research. Besides, I'm involved in designing different research projects using publicly available data sets. So in continuation of my liver cancer research background, here I'm working on the project involved multi-omics data integration of liver cancer patients to find out molecular signature that are associated with clinical and demographic phenotypes of the liver cancer patients. Where we not just use transcriptomics data, but we have also explored miRNA and clinical data that can be integrated in a multi-omics data integration approach. Here we implemented various multi-omics data integration tools like SNF, iCluster Plus, O2 Plus, etc. that are integrated on T-Bioinfo server that has already Ilya has shown you. So finally, we are trying to understand how multi-omics data integration analysis can provide us the holistic picture of this lethal malignancy and also how they are associated with diverse clinical phenotypes like tumor stage, risk factors, race, age, and gender of the patients. So participants can explore this project during this program to get a better understanding. I'm looking forward to being your mentor during this upcoming OmicsLogic Transcriptomics program and will help all of you learn about data analysis, machine learning, and visualization in application to biomedical data sets. So now I would like to pass this session back to Ilya. So over to you, Ilya.
Thank you, Harpreet. So as you can see, the ability to work with data and ask biological questions really has to be done in the context of projects. And another person who's going to be guiding the participants in project selection and making the most out of this program is Dr. Mohit Mizunda. So Mohit, I would like to ask you to share a little bit about how this is going to be done in this program.
Sure, Ilya. Hello, everyone, and thank you, Dr. Harpreet and Ilya, for this wonderful overview. So my name is Mohit Mizunda, and in my current role, I'm working and collaborating with universities to introduce innovative curriculum on data-driven biological research at a high school, undergraduate, and postgraduate levels, and developing bioinformatics research projects. So one of the topics that we wanted to address today, and one of the main challenges that a research project presents is how to get started, right? So for a number of years, our team has been working on project-driven bioinformatics training programs that are designed to help you find, develop, and complete such research projects. So as a result of this experience, we want to offer you several suggestions to make so that you make the most out of your research project. Transcriptomics, which encompasses assessment of alternate splicing, alternate polyadenylation, identification of fusion transcripts, exploration of non-coding RNAs, transcript annotation, and discovery of novel transcripts, right? That's a lot. So hence, we can surely say that analysis of RNA-seq data is a valuable tool for understanding cancer mechanism and identifying biomarkers. So in this program, we will work on this exciting, So in this program, we will work on this exciting project that I wanted to show you. In this project, we will use translational research and biomarker discovery, where we will be looking at cancer subtype characterization, associating technical skill sets that we learn, that how to process the data, eliminate technical issues, and use machine learning tools to extract meaningful insights from complex molecular data sets. So another project that will be a part of this entire training. So this project was inspired by Demian et al. So modeling precision treatment for breast cancer. So this is an analysis of over 70 different breast cancer cell lines and over 90 different therapeutic agents. So this project include SNP data, so microarray data, one type of microarray data, RNA-seq data, so the whole transcriptome data, exome data, and also genome-wide methylation data, which is the epigenetic data. So we discussed about the TCGA project, and another set of specialization are the infectious disease related projects. So the application of transcriptomics data is actually providing pivotal information in infectious disease research. So for example, host transcriptomics in TB and other diseases is a powerful approach for discovery of immune signatures. So for example, if you take transcriptomic analysis of human samples, that can provide insights into which genes are expressed in a sample tissue. And by comparing such transcriptomic profiles in samples from subjects with a disease versus subjects without a disease, so it is possible to identify genes that differs in their expression between the groups, and thus it provides us a picture of a disease signature, right? So and one of the major advantages of learning from the examples that I showed you and then hands-on exercises, so you can quickly adopt and work on your own independent projects. So these interesting research projects are proposed by our previous participants from different programs by utilizing the public repositories. And in this program, we will learn how to navigate through different specialized repositories like NCBI, UniProt, TCGA, and several other databases. So we have been collaboratively working with several universities and reaching out to others to help implement data-driven project-based learning that enables students at a young age to ask significant biological questions, and also to faculty to leverage their knack of research. So we have been working on several bioinformatics projects with these faculties. So our collaborators are effectively utilizing this technology not only for education and training, but also to develop collaborative training programs and funding proposals to support student research projects. So Dr. Harshita here specializes in botany and wants to contribute to agricultural research using bioinformatics tools, investigating the genetic data from tomato. Another example here is from a student. So many proposals from group of students are really have been really very exciting. For example, a group led by Sarah here, she wishes to find out about specific molecular pathways that are responsible for this amazing stress adaptation in tardigrades. Another example of such study is by Arnab Banerjee from JU, who wants to study the lipid biosynthetic pathways in a plant called Simplochus. Simplochus. So the idea is to find out significantly differentially expressed genes responsible for lipid production, so that can help in biotechnology. So the application varies from agriculture, biotechnology, infectious diseases, cancer, and many of our previous participants had this chance to work on these different research projects and present this analysis in reputed conferences and workshops and publish online. So that's about the projects and the research that we have been doing and contributing to. And to tell you more, I would like to invite Deepsha about the program. So over to you, Deepsha.
Okay, thank you so much, Dr. Mohit. I will be taking everyone through the registration process. And as I'm going forward, I see that there are some questions in the chat, whether you receive a webinar certificate for this one. We do not give out a certificate for this particular webinar, but all of our programs and a few of the free resources that I will be introducing you to today will have certificates of their own, which you can take after this program is over. So to join the upcoming transcriptomics program, this is the link. We will also email you all this link via email if you're already registered and joining us. So if you go to this website, you will be able to see details about this program. Okay, Nidhi has a question, how frequently these programs are designed. So Typically, these different multi-omics programs are repeated once or twice a year. So as I mentioned before, the transcriptomics program is being repeated for the third time this year. And of course, in the next upcoming year, we will have the program again. Though right now we do not have any dates for it yet, but we continue to repeat these programs because these are very basic stepping stone kind of introduction for students. Yes, Mansi, all of it is related to bioinformatics. Everything that you see here, these are all related to bioinformatics topics. So this program specifically today is related to transcriptomics, RNA-seq analysis. And we also have programs related to metagenomics or microbiome data analysis. We have programs on infectious diseases. So there are different programs that we do all throughout the year. Yeah, sure. So we will reach out to you regarding the presentation or the video of this program later on via email. Now, just to show you how you all can register again, as I was pointing out. Yes, I will come to that in a moment for the pricing in INR. So as you see in this page, you'll be able to get introduced to this program and you can see the entire syllabus. So we are starting the program next Wednesday, December 9th, and the program will have live sessions until December 23rd. The program does not end on December 23rd. These are the duration of the different online classes or online sessions like today. And after December 23rd, you will have about 2 to 3 weeks until January 9th to work together to complete all of the different courses that are there, go through the different resources, as well as if you want to work on your own individual project. Now, I would quickly want to go ahead and address some of the questions that are there in the chat and then move forward from here. So to know about this program, if you click on Get Started, we offer 3 different levels. If you are a beginner or if you're somebody who is just starting for the first time and you're getting introduced to bioinformatics, yes, if you have not studied bioinformatics, you can still go ahead and join the program. And the reason is because we will be having classes like this where you'll be able to interact with the mentors you met today and who spoke to you previously. And they will answer those questions for you. We have courses. I will give you an example of those courses where you will be able to go through these different materials, which are very, very basic and takes you right from a very introductory level to a more advanced level. So yes, you would be able to do that, but of course, it also depends that you are able to make time for it and give time to it. So that is there. So this is the beginner session. So if you're just joining the program for the first time, then all you need to do is you can sign up for this. So 150 USD is in INR exactly is equal to, okay, I would have to check this up because I'm not really sure, but let me just pull it up for you. Okay, so it comes down to around 11,000 INR. And we will be getting back via email. So if you have any other exact questions, like how much these pricing comes to in your value, you can just let us know via email and I will be responding, because it's not possible to tell to everybody because all of you are joining from different countries around the world. So this is the standard pricing. And here are the payment options. So you can check out via here. And moving on. So what do you do once you are signed up? So how do you get access to these different resources? So the first resource that every person who joins this program has access to is an organization page. So the organization page is like what you can call a classroom. Every member of this program will be part of this organization page. And you can only get access to this page once you're registered for the program. So when you come down, you'll be able to see all the different events for the program. So see, we are starting on 9th December with the overview of the program. Then we go with 11th December with processing of gene expression data. We are going into more details. Then we are exploring gene expression tables and learning about statistical analysis on 14th December, and so on and so forth. So you see there are sessions dedicated to learning about machine learning and single cell transcriptomics, as Ilya was talking about earlier. So you will be able to go through all of these things right here in this one page, which you will have access to, so that you can access all of your resources right from there. So one of the resources that you see here is a forum. So I'll just give you an overview of what a forum looks like. So basically the forum will have several different topics. So this is an example of an old forum that I wanted to just show you. So each of you will be able to post questions, get those questions answered right here in the forum. In case you are missing a session, or if you have questions that you don't get answered, or you're going through the courses and you have these doubts and we are not meeting at that point of time, you can just post on the forum and one of our team members would be able to get back to you. So we also have another question here. Do we have to do the program live? Yes. So as I showed, these scheduled classes are going to be live sessions, but they're also going to be recorded, like we are recording it today. So in case you're not able to make yourself available for the scheduled classes, you can also take this entire course asynchronously, wherein we will be emailing you the video recording of the sessions that you can see. And along with it, you will also be able to go through the different courses that are available. So there are different courses, which Ilya also showed you. We have Transcriptomics 1 to Transcriptomics 4, and there are also other several different courses that you can take. So you will also be able to take these courses asynchronously as well. And if you have any questions, we also have the option for one-on-one mentor sessions, where if you have a lot of questions and you have progressed really well and you have certain in-depth questions or some problems with your assignment, you can actually email us and request for that session as well. So that is also available in case you want to go forward with it. And now I want to show you what your profile will look like. So this is an example from a participant who recently completed our last transcriptomics program, which we had in September. So as you see Tapasree here, she has completed the program. She has over 14,810 points. And the interesting part about these points is the fact that these points are collected each time you do a course. Any of the courses that you do here will earn you around 500 points. And Ilya also showed you the server T-Bioinfo platform. So the more pipelines you run on the server T-Bioinfo platform, you will be getting points for them. Also for the platform where we teach about R and Python, the code Omixlogic, which is right this one, you will also get points for this. So everything is associated with your profile. So you are not only able to do the program, but you can actually see your progress right from day one to the last day of the program. And if I can just show you for Tapasree, you can see the number of courses she has completed during that time period, the number of pipelines she has run right here. So this is exactly how you can also track your own progress, and we can also see whether you are able to progress further. So if there is any problem, you can actually get back to us and we can communicate with you and help you sort that out. So that is the support that we provide for all of our programs, whether you're joining it in the next week or in the next year. And besides these resources, as of course, you will have educational access to this platform, which is our data analysis platform. And we will actually go on step by step through these different logic of analysis, like what is the utility of these different pipelines. And there are different assignments as well within the courses, which will help you to look at the different data sets and work with these different types of multi-omics logic and analysis. And of course, as I mentioned, you will also have access to the code Omixlogic playground as well for learning R and Python, as Ilya has shown you before. So do you all have any other questions that I can help answer? If you have any questions, please let me know. I can take a moment to answer them. If you do not have any questions right now, okay, yes. So Andrea has another question. Can you buy courses separately or is it per month only? So for this program, you do not have to buy the courses separately. Like all of these resources will come under whatever you're paying for the program right here, which is the $150, which comes with both the courses, resources, also free access to all the premium courses which are available on the Omixlogic, and the mentor support, as well as the project. If you are attempting to do the project, then also it covers it all. But if you are interested in exploring or taking up a course on its own, I can send you the link for that after the session. So if you can leave your email in the chat box, then I will be able to reach out to you, and you can also take up each of the single courses, but only that those courses would be self-study ones or asynchronous courses, and it does not come with mentor support. But definitely the courses are very, very detailed, so you would be able to go through them yourself. Do we have any other questions? And thank you so much, Sonder, for sharing this. The project is a publishable project. Okay, so I will show you some project examples that we have from our previous participants who were part of the program. So to publish a project, as you know, takes a long time, and this program is only a 1-month program. But if you're interested only in working on an RNA-seq project or any other project for that matter, we also have a research fellowship program that you can actually take a look at. So that research fellowship is offered for either 3 months or 6 months, and it will be helpful for you if you are thinking of doing a project in the long run. Okay, I hope, Andrea, that answers your question. And if you need more information, I can email you that as well. For Pratik, do we need to know the computer languages for these courses? So no, Pratik, you do not need to know any computer languages for any of these courses, because everything is built for you right here in the cloud. So it does not require you to know the programming languages. If you already know, then that is really, really great. But in case you do not do, there's no reason that you have to start learning it before the program or something. You can actually take these courses without having any prior knowledge of, say, R or Python, or even about algorithms in bioinformatics, which you will see in the different pipelines that are shown out here. So this is why the platform is a very, very student-friendly and great for beginners to start with, who are not very familiar with the more advanced ways of bioinformatics analysis. Okay. All right, I will send you the info, Andrea. Thank you. And thank you, everybody, for sharing your email IDs. So if you all have any questions beyond the session that you would want to inquire about, I'm available at marketing@pine.bio. And you can also reach our team here, marketing@pine.bio, for these questions. If you all have any other questions, I think we have a few more minutes, like 2-3 more minutes before we will conclude today's session. You're welcome, Zore.